very much. Yes, I'm Tom. I'm the coordinator of the IT and Infrastructure Committee. I'm going to talk a little bit about some initiatives going on across ERGA as a whole, but very much with either IT or infrastructure core to its heart. And this is very nice for me as an individual because it means that I'm not going to talk at all about my own work, but really showcase the work of other people. So Louise, if you could show on the next slide, I've got a lovely list of all the people whose work I'm going to try and showcase today. So starting with Felix, Astrid and Rita, who have very much helped on the sample metadata and metadata deposition side. Olga, who's helping us with protocols, curation and publishing. Solen, Diego, Valentina, Jessica, and Sagan, helping us on the bioinformatics side of that, so how we can collect, curate, and publish bioinformatic workflows. Tyler, who's just about everywhere in ERGA, so I don't want to pinpoint him to one project. And Mathieu and Beth, who are helping us at the end of a project with how we can actually publish our results in a consistent and ergified manner. So Louisa, next slide, please. I call this talk Aligning ERGA with the FAIR principles for data and metadata and metadata. And I want to just introduce this concept that metadata and data should be findable, accessible, interoperable, and reusable. And this is not just a vague concept which is handed down on high, but really one that we should think about when we're handling our metadata, our data, and our whole research, as it helps us as a community interact with each other. We know that our data are consistent, that they are understandable with each other. Our metadata are rich and describe what we're producing. And then we can transfer this to others as we hope to expand as a community. We know that what we've created is understandable, it's reusable, and it's interoperable. So our aim is to be as open as possible, but as closed as necessary. And I'll come on to why this is important later. Our data and metadata should be published openly where appropriate. The means by which we've obtained our data and metadata should be open and also understandable. The vocabulary we use should be understandable and approachable. And if we are consistent in all these steps, it also means later on that when we come back to meta-analyze our work and learn and move forward, this is a much easier task for us down the line. So I've highlighted at the bottom different stages of the protocol that I'm going to touch on today. So it's in the metadata, the secure data, our protocols and workflows, the publishing and collection of genomic data, and then finally how we publish our results at the end. Next slide, please, Louisa. So our metadata that we collect should be, as I said, understandable and interoperable, both in terms of the language, but the fields, and that means to both a human, but also a machine. The metadata we use should be rich in the description of the data and compliant with the standards of our community as we move forward. And these concepts are quite broad and perhaps inappropriate at the start, but the SSP committee, with particularly Rita and Astrid, along with COPO, have designed a sample manifest, which, when filled out, means that you are already compliant with these ideals of being accessible, interoperable, findable, and reusable. And this is great because it just means you have to, if you know everything about your sample, you can find the sample manifest, you can fill it out, and you know that COPO will handle the rest of it and making sure that all of your metadata are compliant and readable in the future. This time, Lisa. Thank you. Now, not all the data we collect as part of this project needs to be open, and this is particularly pertinent to permit material transfer data, which should not be visible to the entire public. So when it comes to these data, we know that these should be secured in a permanent archive location, and they should be indexed and findable, meaning that when there are locations where perhaps we're audited or we're asked about the provenance or the legality of our sample, we know we can go back and find it in 1, 5, 10 years' time without having to know which individual placed what file where on a folder. So with this in mind, we're working with another Horizon-funded project, EUDAT, and the COPO team to store all permits, MTAs, documents linked to samples in a unique indexed way, so that in the future we know that even if all of us leave the project and hand it over to future people, people can find these documents when required. And this is really important. And a key point of the FAIR principle is that findable means findable to the relevant people, not to everyone in the world. Thank you, Louisa. So there's no point generating all this metadata and data if we can't explain how we've generated it in the first place. And we want to be able to show to the public that we can explain this in a way that others can reproduce in the future. So our protocols and workflows should be state of the art, and this will change over time. We want them published in a location that can be easily accessed, written in a way that not everyone, but at least appropriate individuals can understand and reuse, and they should be permanently indexed and archived. So on the sampling, lab-based and sequencing side, Olga, as I mentioned, is helping us set up our space on protocols.io, where we will collect, collate, and curate sets of protocols, which can then be published under an ERGA and BGE umbrella, meaning that these protocols will then be findable in the future. And once published, they can even be referenced to make writing method sections much less painful in the future. On the informatics side, we have a whole team of people trying to do the task of writing assembly, annotation workflows and publishing them in either Galaxy, Snakemate, or Nextflow workflow languages, and publishing these on Workflow Hub. And once again, once these are published, they're frozen with DOI and they can be referenced in the future and reused for future publications, and means that we can pinpoint exactly which pipeline was used for which assembly or which annotation moving forward in the future. Thank you, Louisa. Once we have all of our data, our genome, our annotation, we want these all to be visible in a consistent and findable way. And this is where the COPO team come in again, and also with Talbot Tyler, into submitting this metadata and this data consistently under one, in this case, bioproject or biosample to the European Nucleotide Archive. So all of this in the ENA will be open and accessible, and under our consistent ERGA umbrella bioprojects, we hope they will be easily findable. And going through these channels, it means that our metadata and our data will be linked permanently and with unique identifiers, and consistent in their layout. Once again, meaning that any metadata, meta-analysis in the future will be much easier, and people will know as soon as they click on a project, I found an ERGA project. I know this is of a high standard. I know I'm going to be able to find my data, my genome, and all my metadata at the standard that it should be. Thank you, Louisa. And finally, once we've done all this, we want to be able to showcase our work to the world. And Mathieu and Beth are helping us to expand on the great work they've done with the Tree of Life in their automated genome notes to help pull together an ERGA branded genome report. That means that we can pull together everything that's publicly available in a consistent and standardized way, whether this be pulling out the metadata, data, all of the outputs, the workflows, the protocols, and putting this in a nice format that can be published and read by a wider public beyond ERGA. Once again, keeping this consistent means that in the future, we can come back and analyze all of our ERGA outputs over the next years. And we think this is very important when we especially reach the end of this project, to be able to look back and see how we can learn, how we can move forward in the future. So I think to my final slide, Louisa. I really wanted to showcase that there's a lot going on centered around this community. And I wanted to introduce these names to you so that if you want to get involved either with the committee or to learn more from these individual people, happy for you to email this address and we can either bring you to the committee, put you in contact with the people, or if you would like to present something that you think is appropriate to this group, please let us know. And for that, I'll say, yeah, so thank you very much to everyone that's contributed to this that I've named and those who I've not been able to. Thank you to Luz for giving me a hands-free experience and for everyone for listening.